thanks for inviting me, and I'm happy to be here. I want to tell you today about a global initiative to create an atlas of the human body, focusing on all the cells that the human body contains and the specialized cell types it contains. I'm currently a professor here at the Karolinska. I studied molecular biology at the Stockholm University. And then I came here a long time ago to do my PhD, where we studied neurotrophic factors, which are proteins that guide brain development and also are involved in learning and memory. But in the last few years, I've been focusing on brain development on a larger scale, trying to understand brain development on a systems level. And I'll show you first some about aspects of the Human Cell Atlas project, and then I'll give you an example of a pilot project that we're running in my lab. So the Human Cell Atlas project is a global initiative. It's an initiative from scientists who have gotten together, several hundred scientists, to take the initiative to create an atlas of the human body focusing on cell types. And a good analogy is actually Google Maps. Because the kind of atlas that we want to produce and need to produce of the human body will need to contain many layers of information so that we can overlay fundamental physiological features such as cell types with all kinds of other information such as specific proteins that are active in specific cell types, or electrophysiological properties in the brain, or mutations involved in disease. So we are envisaging a three-dimensional atlas of the human body with many layers of information that can integrate our knowledge about the human body. And the atlas, as I said, is focused on cells and cell types. And the reason for that is that cells are the basic unit of life. Cells are what makes up all of our organs, of course, and cells are the carriers of the genetic information. But they're also the functional unit that executes and interprets the genetic information in specific tissues. And the reason that tissues are different is because different tissues contain different types of cells, as illustrated here. So there are many kinds of cells in the human body. And in a sense, the Human Cell Atlas project began more than 100 years ago. People have studied cells and tried to classify cells for many, many years. And traditionally, mostly based on shape, because that's what you could observe using light microscopy. And you can get quite far. You can see that all of these specialized cell types here in different tissues, they have very characteristic shapes, as illustrated by these cartoons. But as it turns out, within classes of cells, like ependymal cells, for example, here, or neurons even more, even though they have same or similar shapes, there are many, many subtypes that perform distinct functions. And in order to learn about those, we need to go to molecules. So we need to understand cells on a molecular level. And doing this analysis on the molecular level is also very important because molecular events in specific cell types are what underlies most human disease. So if you get a mutation affecting DNA, this same DNA is in every cell of your body, and the mutation will be carried to every cell in your body. But different diseases will only manifest themselves in specific tissues, such as, for example, autism in the brain, because the gene that's mutated is active somehow in neurons or when neurons develop during embryogenesis. So it's only if we understand all the different kinds of cells that exist in the human body and understand which genes are active specifically in those cells, in those cell types, it's only then that we can begin to understand human disease on a truly molecular level. And the problem is, we do not actually know our cell types. The human body is estimated to contain something like 30 trillion cells. Textbooks will tell you that we have something like 300 cell types. But recent studies have shown that if you look very carefully using the latest molecular tools in a tiny piece of tissue like the retina, you can actually distinguish at least 100 subtypes of neurons in the retina. And these are neurons that have specialized function, for example, detecting lines in particular orientations, or detecting contrast, and so on. So they really have a function in the retina. And if there's 100 subtypes of cells in the retina, then obviously in the entire body, there must be way, way, way more than 300 cell types. So there's a huge discrepancy between our current sort of accepted understanding of human cell types and the reality that we do not yet know. So imagine that we could make a map of all the cell types in the body. What can we learn from such a map? Well, first of all, it would be a reference of the healthy human being. So it would be a template against which you can compare, for example, genetic variants, and distinguish and compare pathological cells versus normal healthy cells. More fundamentally, in terms of biology, it will start to give us an understanding and a definition for cell types and states and transitions and lineages, all of which are currently very vague concepts. But I'm convinced once we have generated these kinds of large-scale atlases, we will begin to understand the structure of how cells are organized into types and classes. And of course, it's a direct view into human biology in vivo, by which I mean a very large proportion of research currently in human disease uses model organisms or cell culture. But going directly to the human body and understanding cells, naturally occurring cells in their tissues, will be much better models of disease. And having a reference atlas of the normal body will also let us assess whether our in vitro systems are actually good models of the true tissue. So if we are testing drugs on neurons that we are culturing in cell culture, we need to make sure that those neurons are actually similar or hopefully identical to the corresponding neurons in the brain. And that's one thing that you can use an atlas for. And then we can use this atlas to understand how the body develops, using understanding the molecular code that underlies cell differentiation and how cells become different during embryogenesis. And as a spin-off, this kind of ambitious global project, will also, of course, generate new technologies and new computational tools. So I'm studying the brain. This is one of the oldest drawings of a brain that shows the cell types, the major cell types of the brain. It's more than 100 years old, and it's a piece of cortex. And you can see here in black are the neurons, and you already see that there are many different shapes. So probably there are many different kinds of neurons here. And in green, you see all kinds of glia cells. These are the supporting cells and the immune cells of the brain. And you also see the vessels in red and blue, which here are drawn just as tubes, but of course are also made up of many different cell types, like endothelial cells and pericytes and so on. And this is already highly complex, but this shows you only a small percentage of the cells that are actually in this picture, because this drawing is a schematic and it's drawn after Golgi stain that actually stains only a small fraction of the cells. So in reality, this tissue that you're looking at here would be packed with cells, completely packed. So, and there would be many, many different kinds of cells in this tissue. And of course, this in turn is just a small part of the brain, and the brain is just one part of the entire body. So it's a very complex system. And in the past, we've been able to study tissues such as the cortex here only by bulk methods, meaning that we would dissect the tissue and then we can analyze the sort of average molecular composition of the tissue. So we can compare cortex to, let's say, cerebellum, and we can detect genes or proteins that are specific to one or the other. But then you're averaging over all of this jungle of complexity. So what has happened recently, which has made it possible now to dream of actually generating a human cell atlas, is the development of single-cell analysis methods, taking analytical methods down to the level of individual cells. So we can actually measure cellular properties individually and at large scale. And the idea is very simple. You take out cells from, for example, this is the somatosensory cortex. You isolate a random collection of cells, so you don't sort them or select them in any way, you just take a random sample. And then one by one, you analyze them, analyzing some molecular property. For technical reasons, the one that is most easy to analyze is RNA, which gives, of course, information about gene activity. So the genes that are active will generate RNAs. And so we can detect patterns of RNAs that are present in each single cell. With the idea that cells of different types will have different patterns of gene activity. And this is a straightforward assumption because different types of cells perform different functions, and to perform different functions, different genes would have to be active. For example, in neurons, you need ion channels, so all the genes that encode ion channels will need to be active in neurons. Whereas in, let's say, an insulin-producing cell in the pancreas, you would need to have all the enzymes to make insulin would need to be active in that tissue. So in that sense, gene activity and specifically RNA serves as a kind of fingerprint for cell type. So assuming this works, then in the computer we can now organize the cells by similarity. So for example, the green cells generated very similar expression profiles here, and therefore we would group them together in the computer. And this way we can identify the cell types that are in the tissue. So I'll show you an example of an experiment that we did recently, where we studied exactly this tissue, the somatosensory cortex here. This experiment here is done in the mouse. In this case, we took somatosensory cortex, we dissociated it, so we got basically a tube filled with cells that are dissociated from each other. And then we used the microfluidic chip to capture cells. So here you see a neuron that's been captured inside of a tiny little channel. This cell here is probably about 10 micrometers in diameter. So you're looking at a very small channel inside of this microfluidic chip. And we capture cells one by one, again at random. And each cell, which contains now RNA molecules that give us information on which genes are active, each cell is lysed to open it up, and then we add enzymes to convert RNA into DNA, which we can sequence using high-throughput sequencing. And thus we can learn for each individual cell which are all the RNAs that are present, and hence which are all the genes that are present. So here's what the data can look like. This is a heat map, and it's like a grid of values. And when it is yellow or red, it means high activity. And when it's blue, it means low activity. We have 3,000 cells. So in this experiment, we generated 3,000 cells here. So each column here is a single cell. There are many, many columns, so you almost don't see them, but vertically you have the cells. And then horizontally we have genes. So for example, here are 800 genes here that are active in a set of 800 cells. So there's a group of cells that is characterized by having these genes active, these 800 genes. And you can see that the whole data set sort of decomposes into these big blocks. And these big blocks correspond actually to all the major cell types that we saw in that drawing from 100 years ago. Neurons, these were the black cells in that drawing. Glia cells, that were the green cells. And well, other types of glia, and also vascular cells, which are down here. So now we actually managed to discover all of the major cell types of the cortex using molecular methods. But since we have individual cell measurements, we can go much deeper. So you see that within the class of neurons, there are many, many genes that are expressed in neurons. Most of them have sort of similar patterns. But there are genes that are specifically more active in certain neurons than others. So there's a lot of structure in here. And this is because there are different subtypes of neurons. So if we take this cluster here, these are, we identify them as cortical pyramidal neurons because they have this triangular pyramidal shape. We take out just that part of the data set. So now we have about 400 cells, and we have about 1,000 genes. And again, now we subdivide this even more. And again, you see this block-like structure. So again, there are genes that are more specific to certain subsets of the cells. Of course, the data is now much noisier. It's much less clear-cut. So we're down to fine distinctions, fine subtypes of cells. So to convince ourselves that this actually corresponds to specific types of neurons, we can now go back to the tissue. So I'm going to walk you through this carefully. Here we have 1, 2, 3, 4, 5, 6, 7, 8 clusters, approximately. We name them like this. So each label here corresponds to one of these. So this box here, these are genes that are expressed in these cells. We label it S1 pure L23. Doesn't matter why, but that's what we label it now. And if you look at genes that are active here, we can find specific genes, RAS-GRFL2, for example, or PVRL3, that are enriched in this cluster compared to all the others, so in these cells compared to all the others. So these are genes that are specific or at least highly enriched in this specific subtype of pyramidal neuron in the cortex. Now we can take those genes because now we learned something about these cells. We learned that they express these genes. And this acts as a molecular handle that we can use to go back to the tissue. And we can actually stain the tissue and see where is this gene active. And it's a bit difficult to see here, but these are kind of strips of cortex like this through the cortex. So this is the surface of the brain. This is the ventricle. And we're staining here for RAS-GRF2. And you can see that cells that are in what is, this is layer 1, so this is layer 2 and 3 of the cortex. Those cells express RAS-GRF2. So this shows that these cells here, which we identified here as expressing RAS-GRF2, they're actually located in layers 2 and 3 of the cortex. In contrast, if you take this cluster down here, it expresses, let's see, which one is it? It's probably this one, complexin 3 here. So it's S1 pure 6b. It expresses complexin 3 and to some extent SULF2. And if we stain the tissue for complexin 3, we see it's not in layers 2, 3. It's actually in layer 6 or layer 6b, so very deep in the cortex. So what this shows us is that we are able to discover, using these molecular methods, discover specific subtypes of pyramidal neurons in the cortex that correspond to layers of the cortex. And this makes a lot of sense because this is how the cortex develops. So the cortex in the embryo develops layer by layer like this. So it makes sense that these layers would also be molecularly different. Okay, so that was one very specific tissue. If we are going to do an atlas of the entire human body, we need to develop methods and analytical tools that can scale to really large tissues and organs. And so we are currently doing a pilot project where we are trying to create an atlas of the entire mouse nervous system. Here is a mouse, a slightly fat mouse, and this is the nervous system. So here's the brain, this is the spinal cord. But along the spinal cord are also sensory ganglia and also sympathetic ganglia that control autonomic functions. And you also have neurons along the intestine called enteric neurons that control bowel movements. So we sampled from all of this. We sampled from the brain. We subdivided the brain into many different regions. And we also sampled from the peripheral nervous system. And we did the same thing. We took individual cells one by one, measured RNA, so we could measure gene activity. And then we arranged the cells in silico by similarity. And it's what we're showing here. We have a total of here something like 350,000 single cells. And if we now focus on one tissue here, this is cortex, it's the frontal cortex. Here, I don't know how many cells there are, but maybe 20,000, let's say. So each little dot here, which you almost cannot see, is a single cell. Okay, and the cells are arranged by similarity, and they're colored by major class. So again, those major classes that we saw in the drawing from 100 years ago. So the blue ones are neurons here, and orange and green ones are glia, and then you have vascular cells, for example, in violet here. So this is very preliminary work, but you can start to see some patterns across the nervous system. For example, you see there's a lot of heterogeneity among the neurons in the cortex, meaning that there's a lot of different kinds of neurons in the cortex. It's a complex tissue. Whereas in the cerebellum, for example, there's only a few types of neurons. It's a more simple structure in that sense, in terms of neurons. You can also see that in all these tissues, which are sort of frontal, so towards the head or towards the nose of the animal, there are very little of these orange oligodendrocytes. Oligodendrocytes are the cells that make myelin, that ensheathes axons and increases the speed of propagation of the action potential. So in the forebrain, most connections are local and short range, so you don't actually need to accelerate action potential conduction. When you go towards the back of the animal, this is the hindbrain and this is the spinal cord, you see this huge cloud of oligodendrocyte. And this is because here you have nerves that go from the brain all the way, in essence, all the way down to your toes. And in order to propagate the signal so far away, these nerves all need to be myelinated. Oh, sorry. If we look here in the middle, this is the hippocampus. It's an important part of the brain that's involved in learning and memory and spatial orientation. You can see a color that you don't see anywhere else, which is red. There's a red cluster here. And those cells are cycling, meaning that they are dividing. These are cells that are proliferating and making copies of themselves. And this makes sense because it is actually one of only 2 places in the brain of the mouse where neurons are being regenerated even in the adult. So this structure here, it's continuously making new neurons. The other place is the striatum, so you have some cycling cells there as well, or technically the subventricular zone. If we zoom in to the hippocampus, you see these cycling cells here, and this is getting a bit abstract, but again, you're seeing all the neurons in blue here. You're seeing these cells that are dividing. These are kind of stem cells for neurons, so they are ready to make more neurons. When they stop dividing, they will generate all of this cloud of granular cells, which is the main cell type of the dentate gyrus in the hippocampus. And if we zoom in, of the dentate gyrus, yes. And just to point out what this cloud represents, so this is so the cells are organized by similarity. So they start here, and as they mature, they sort of move along here. And just to explain what this means in terms of tissue, in the dentate gyrus of the hippocampus, so this is this part here, this is the hippocampus, and there's a dentate gyrus. Here are the stem cells. They sit along the edge of this little blade. They divide and they go through a few steps of differentiation and integrate and become these granular cells. So this is what we observe in terms of molecules and gene expression, is actually this process of making new neurons in the brain. Okay, so I will end with a few words about the second part of the atlas. So far, we've talked about classifying cells based on gene activity. But this gives you basically just a list of cell types and molecular properties. But to make a real atlas, you need to have spatial information. You need to know where the cells are. And since we are defining cells by gene activity, basically by the presence of specific RNAs, in order to locate cell types specifically in tissues, we need a method to detect RNA in tissues. So one such method is called single molecule FISH. It stands for fluorescent in situ hybridization. And it's a method where so RNA are this this line here depicts one RNA molecule. So RNA, of course, for a specific gene, the RNA has a specific sequence. So we can target this using short DNA probes that will bind to this RNA. And here I'm showing 3, but in reality, we target using 25 or 40 probes. So if a particular kind of RNA molecule is present in the tissue, and we throw on these probes, something like 25 to 40 fluorescent probes will come together in a tiny spot that is just one molecule. And so if you have 25 to 40 fluorescent molecules in the same tiny little spot, if you look in the microscope, this creates a dot that you can actually see. So it's a little bit hard to see here, but all these green dots here, for example, they are mRNA molecules, individual mRNA molecules. You're seeing actually in the tissue individual mRNA molecules. And these molecules are from the gene SLC6A11, which is characteristic of one particular cell type that we're interested in. And by doing this over and over in different colors and also cycles, so we can stain for a few genes and we can wash and we can stain for a few other genes in the same section, we can create a composite image that shows the presence of many different cell types. And let's see if you can see this. Yeah, so this is again the cortex, our favorite. Here's the surface of the cortex. Here down is the ventricle. And in green and blue you see cell bodies and cell nuclei. So green is the cell bodies and blue is the nuclei. And we can do an image analysis on this image in the computer and segment it. So now the computer has identified for us where all the cells are and just made them in different colors so that we can see that they have been identified. If we zoom in, this is almost impossible to see. Is it possible to turn off that spotlight? Just briefly. So here you see outlines of cells. And inside of the cells you see dots that represent mRNA molecules for specific genes. So here is a cell that expresses two different genes that we have probed, one that is labeled in white and one in red. And here's another type of cell that's labeled only with the yellow probe. So this I know it's an oligodendrocyte, for example, because we stained it with PLP1, which is an oligodendrocyte gene. So in this way we can locate specific types of cells simultaneously in the tissue. And if we do this at very large scale, sadly we don't see anything, but this is the cortex again, and this is a much bigger part of the cortex now. Here is white matter where you see oligodendrocytes, and in here are all the specific cell types that I've listed here. Let's see if we zoom in, if we can see anything. Okay, slightly more. So now what we're looking at here is, again, the cortex with the surface up here. If you look carefully, I think you will see red staining, for example, here and here and here. Those are the vessels. And you see blue staining all over here. Those are the neurons, the pyramidal neurons, excitatory. You also see this magenta staining. Those are inhibitory neurons, also called interneurons. And you have pericytes, which are also vascular cells in orange. So now we are back to that image from 100 years ago. But this image now is not a drawing. It's actually data that shows gene activity in specific cell types, all simultaneously in the cortex. So our dream is now to do this for the entire human body, as part, of course, as a large international consortium, to create an actual atlas of the human body, a cell atlas. And I will end there to thank all the people that we worked with, my group, all my collaborators, our funders, and the core facility that helped to do all the single-cell analysis. And if you're interested in this kind of stuff, we are recruiting postdocs right now, and we're also always interested in master students. So, thanks.